So our next section for in biochemistry is laboratory techniques. So first we will discuss about polymerase chain reaction or PCR. Definition: it is a molecular biology technique that is used to amplify DNA fragments. Uses a diagnostic tool, for example, neonatal HIV and herpes encephalitis. Its definition is it amplifies DNA fragments. Think of it like a photocopy machine that is a photocopier for DNA. It is a diagnostic tool for neonatal HIV and second for herpes encephalitis. So steps for PCR. So we will remember there is a mnemonic for steps. Remember steps of PCR by a mnemonic, that is D, A and E. For D is for first step is denaturation. Second step is annealing. Annealing, its also meaning is binding. Third step is elongation. Denaturation, DNA template, primers, heat stable DNA polymerase and dNTPs are heated. So these are components of DNA. So DNA template, primer, heat stable DNA polymerase and dNTPs, these are all DNA components. They are heated at very long temperature. Due to extreme temperature, heat separates the double-stranded DNA into single strands. So heat separates these double strands into single strands. So first step, denaturation process is done, and we have got a single strand that is it has very high temperature because of heat. So second step of PCR takes place, that is annealing. Annealing is basically binding. So first sample is cold. After denaturation, that sample we got, that sample is cold. Primers bind or anneal to specific DNA sequence to be amplified. Specific DNA sequence to be amplified. Elongation. Temperature is increased again. So in elongation process, during the elongation process, temperature is again increased. DNA polymerase adds dNTPs, extending the DNA strand after each primer. This cycle repeats, heating and cooling, until enough DNA is produced. So this was PCR. PCR is the goal for detecting viral DNA, RNA in clinical samples. So we have discussed about the PCR, that is very main lab technique. That is also a diagnostic tool for neonatal HIV and herpes encephalitis. Now we will discuss about CRISPR or Cas9. Definition: a genome editing tool that is derived from bacteria. So CRISPR Cas9 is a genome or genes editing tool, and it is basically derived from bacteria, and its components are guide RNA or gRNA and Cas9 endonuclease. So these two are its components. Guide RNA, gRNA, that is complementary to target DNA sequence, and Cas9 endonuclease, it cuts DNA, single or double strand break. So Cas9 basically it cut double strand DNA into single strand, or even it also cut single strand DNA. So it's the main function of Cas9 endonuclease. Discuss about application of CRISPR Cas9. So first, it removes virulence factors from pathogens. So it is the first function of CRISPR Cas9. It replaces disease causing alleles with healthy variants. For example, in the sickle cell disease, it replaces disease causing allele with the healthy variants. It also targets tumor cells for therapy. It is the third process. It targets tumor cell for therapy. So these are three applications of CRISPR-Cas9. Outcomes. There are two outcomes of CRISPR-Cas9: knockout and knock-in. Knockout: frame shift mutation that leads to gene inactivation, and knock-in: edited sequence inserted via donor DNA. So these two are outcome of. Now we will discuss about the blotting procedure. One is southern blot. Steps. In southern blot, DNA sample is cut out into the fragments. That fragment, these are separated by gel electrophoresis. First, DNA sample is cut into the fragment, and these segments are separated by gel electrophoresis. That leads to transfer to membrane. Second step is membrane exposed to labeled DNA probe binds complementary sequence. Third, visualize using film or digital image. Uses of southern blot: it identify size of DNA sequence, detect heterozygosity, and it is southern blot is also used for diagnosis of Fragile X syndrome or CGG repeats in FMR1 gene. So these three are uses of southern blot. So here is a mnemonic to remember. Blotting techniques. So mnemonic is snowdrop. S is for southern, N is for northern, O is silent, and W is for western, and D is for DNA, R is for RNA, O is silent here, and P is for protein. So mnemonic is snowdrop. Snowdrop is its mnemonic to remember this. Now we will discuss about northern blot, that is for RNA. Northern blot, it is similar to southern blot, but it uses RNA samples. Uses of northern blot: it study mRNA levels and size. mRNA level and size, these are studied by northern blot. That leads to reflects gene expression, and it detects splicing errors. So these are main uses of northern blot or RNA blot. Western blot or protein. Steps: protein sample separated by gel electrophoresis. In Western blot, protein sample is separated by the process of gel electrophoresis. Then that sample is transferred to membrane. Labeled antibody binds to specific protein. Uses of Western blot: it identifies specific proteins and it determines protein quantity. Key takeaways: PCR, it amplifies DNA, that is used for diagnostics techniques. Where it is used, PCR diagnostic techniques for PCR, it amplifies DNA and it is used in diagnostic techniques. Where it is used in diagnosis of neonatal HIV, and herpes encephalitis. So these two are diagnostic uses of PCR. CRISPR or Cas9, it is a genome or genes editing tool. that is knockout and knock-in. Now we will discuss southern blot. Southern blot is used for DNA analysis. Northern blot. Northern blot is basically used for RNA expression. Western blot. Western blot is used for protein detection. So these are the three uses of blot techniques.